Hello students, myself Saurabh Palekar, and today I am going to talk about the study of mitosis. So let us move on with the experiment. The aim of the experiment to study the stages of mitosis using permanent slides. Principle: the mitosis is an equational division in which a parental cell produces two daughter cells that resembles the parental cell in terms of chromosomal number. So this maintains the constant number of chromosomes in each cell of successive generation. It occurs in somatic cell of the body, so it is also called as somatic cell division. This means what? The mitosis can be called as equational cell division because in a parental cell and a daughter cell, okay, if you compare, you will see that number of chromosomes will remain same. Okay, it will not vary. Like for example, if you take a human cell, the human cell will be having 46 chromosomes. That means it is 23 pairs. So if a cell is undergoing a division and producing two daughter cells, if you count the number of chromosomes, the number of chromosomes will remain same in both the parental cell as well as in the daughter cell. Yeah, so this is how this is why the mitosis will help in maintaining the common what is it, constant number, okay, of chromosomes, generations after generations in the these cells. So since this division, okay, it occurs in the body cell, okay, that is, it's also called as a somatic cells, okay. So this division is also called as a somatic cell division, okay. So moving on to the requirements, the it the in order to identify a particular stage, okay, you should have a permanent slide, or you have to make a new slide, a temporary slide, wherein you can observe the stages, okay. So observation, record the observation and draw the label diagram. So you need to observe the stages and draw the diagram. So before we move on with the concept, you should know the entire cell process, how which stages are going to be present or involved in it. So the mitosis, it basically is divided into two parts, okay, or two phases we can call. One is called as the interphase, and second one is called as the mitotic phase, also known as the M phase. The interphase is further divided into three substages. One is called as gap 1 phase, or also called as G1 phase. Next comes the synthetic phase, also called as S phase. And third is gap 2 phase, also known as G2 phase. Okay. So once this interphase gets completed, okay, after G2 phase, the cell will enter into the M phase, or also called as mitotic phase. Now this mitotic phase will be divided into two types. So first, it will undergo the nuclear division. That is what we call it as a karyokinesis. And then comes the cell division or cell cytoplasm division. We call them as cytokinesis. Okay. So first, initially, the karyokinesis has to get over, and to do that, it has further four substages. Those are nothing but prophase, metaphase, anaphase, and telophase. Okay. So once this telophase gets over, that means that there is the completion of nuclear division within the cell, and then it can enter into the cytoplasmic division, wherein two daughter cells can be formed in the end of this mitosis. Yeah. So this is how the one mitotic cell will divide into two daughter cells. Clear? So let us study each phases step by step. So first comes the interphase. Okay. So inter refers to in between. So it is a phase between the two mitotic cell divisions. Clear? So in this stage, it is a long metabolically active phase between the two successive mitotic cell divisions. This means what? Here initially it was assumed that this was a resting stage of the cell because it was not exhibiting any activity. But later on, due to further research, when it was being made, the scientists found out that it was not a resting stage, but rather it was a preparatory phase. So this was a stage where the cell was gathering all the nutrients and all the requirements for its further cell division process. Yeah. So this stage has three substages. That is gap one phase, okay, wherein it tries to gather all the nutrients and all the content for its cell division. Then in S phase, which is a second phase, a second subphase. So in S phase, also called as synthetic phase, here you will find the cell will undergo duplication or replication of DNA. Okay, so the DNA gets doubled in this phase, as well as in the cytoplasm, the centrioles of animal cell, it also get duplicated. Clear? Then after synthetic phase, the cell will enter into the G2 phase. Okay, G2 subphase of interphase. So G2 refers to gap 2 phase. So here there are certain proteins which are required for the production of spindle fibers. Okay, so for them, for that purpose, the G gap 2 phase is very important. So once the gap 2 phase gets over, this completes the interphase and the cell will be ready to enter into the M phase, also known as the mitotic phase. Okay. So in M phase, it is basically divided into two types. One is called as karyokinesis, another is cytokinesis. So the word karyon refers to nucleus, kinesis refers to movement. Okay, so nuclear movement will be called as karyokinesis, whereas in cytokinesis, cyto refers to cell and kinesis refers to movement. So cytoplasmic movement will be called as cytokinesis. Okay, so let us study the karyokinesis and then we'll move on to the cytokinesis. First stage, okay, of karyokinesis is nothing but a prophase. So pro refers to before, okay, or initial phase, okay. So phase refers to appearance. So initial appearance is nothing but we call it as a prophase. So here in this stage, what happens? So you can observe this is a animal cell and this is a plant cell. Okay, so there is little differentiation. Okay, so now in animal cell, in prophase, what happens? So during this phase, the chromatin, okay, which is chromatin means the chromatin network. Okay, it is organized into distinct chromosome by coiling or spiralization. This chromatin network, they come together, they get coiled to those nucleosome what we say nucleosome proteins, and they form, that is octane proteins and the histone proteins, and they form the chromosomes in the end. Clear? So this occurs in the nucleus. At the same time, the centrioles of animal cell, in plant cell there are no centrioles, okay, but whereas in animals there are the centrioles, they get converted into the esters, whereas in plant cells directly the esters are going to be formed, okay, and the centrioles are going to be formed and they move towards the opposite poles here. Okay, so these are the centrioles which are moving to the towards the opposite poles, and they starts producing the ester starts producing the spindle apparatus. Okay, or you can call them as spindle fibers. Clear? Then at the same time, slowly the nucleolus, okay, and the nuclear membrane starts to disappear, and the chromosomes are set free into the cytoplasm. So in this stage, you will not find the late prophase stage. You will not find any nucleolus nor nuclear membrane. Okay. So once this stage has been completed, it enters into the next stage called as metaphase. Okay, so before it enters into the metaphase, the spindle fibers which are present, they try to reach towards the center and reach and eventually end up with the connection between the two poles. Okay, and they are also connected with the chromosomes. Okay, so this second phase, okay, called as metaphase. Okay, so meta refers to after. Okay, so it's after the prophase stage. Okay, so here what happens? The chromosomes are completely formed. See? Okay, so the chromosomes are completely formed, and even the spindle fibers are completely formed. So here the chromosomes become short and thick with two distinct chromatids. So you can see X-shaped structure. That means there are two chromatids in one chromosome. Okay, so here all the chromosomes, they move towards the center of the cell and arrange in an equatorial plane. So that means these chromosomes, they are arranged perpendicular, that means right angles to the spindle fibers, okay, and they're present at the equatorial plate. Equatorial plate means in the sense in a line in towards the center of a cell, they are going to be arranged, okay, and this plate is called as a metaphasic plate. So how these chromosomes are going to be attached? So these spindle fibers, they are attached at the centromeres. Okay, so here at the center, okay, you'll find the centromere, that is the primary constriction which you observe, that is a major groove or a major depression which you can observe here. So that is what we call it as a centromere. So they will find the centromere, and to that the spindle fibers are going to be getting connected, and all these chromosomes, they will be aligning towards the center. So this is what will be considered as a metaphase. Okay, once this metaphase occurrence is completed, it will later end into the next phase called as anaphase. So here in anaphase, the ana refers to up. That means the chromatids which are present, okay, they are supposed to be moving upwards towards the poles. Okay, hence the term anaphase. Yeah. So now the centromeres of all the chromosomes, they undergo longitudinal splitting. So that means the chromosomes, they will undergo longitudinal splitting, and the each chromatid, okay, of that particular chromosome, they will be entering towards the opposite pole. Okay, so they will be entering towards the one particular pole. Okay, so the daughter chromosomes, they move towards the opposite pole from the equator by the activity of spindle fibers. That is what is a peculiar characteristic of anaphase. Once this is done, the cell will enter into the next stage called as telophase. So telo refers to end, phase refers to appearance. So telophase can be also called as reciprocal of prophase. It is inversion of prophase. So during this, the event of prophase are reversed. That is what I told now. The daughter chromosomes, it it reach the opposite poles. Okay, the chromosomes starts to despiralize. Okay, so this chromosome starts to despiralize, and it starts forming long thread like structure. That is what we call it as chromatin or chromatin network. Then the nucleolus and nuclear membrane, it starts to reappear. Okay, so and at the same time, the spindle fiber which was been formed, the spindle fiber starts to disintegrate. So this is this condition where the cell is still the cell is still in one, it is, we can call it as one cell, but it has two nucleus. Okay, so it is a two nucleated condition which we can consider. Now it has to undergo the now it has completed the karyokinesis, and it will be entering into the cytokinesis step. So cytokinesis refers to cell movement. Cyto refers to cell, kinesis movement, meaning movement. So the cytokinesis in plants and animals both are different. So this is a image of the animal cell. Okay, so in animal cell, what happens? It is a division of cytoplasm, obviously. So a cleavage furrow develops in the middle of the cell in a centripetal direction. Okay, due to the constriction of microtubules. So there are certain components like microtubules. What they do is they try to pull the plasma membrane inward. Okay, so a inward pull has been occurring, so that is what we call it as a centripetal force. Okay, or we can call it as centripetal direction. So here what is going to happen is a constriction is going to be occurring. Okay, say depression is going to be formed. That depression enters inside from both the end, and it reaches to a point, it reaches to each other, and that completes the cell division. Okay, so the plasma membrane gets divided at that point. Okay, so this is how the cell division occurs in animal cells. Clear? Whereas in plant cell, what happens? The cytokinesis occurs by the formation of phragmoplast. So phragmoplasts are nothing but the cell plates, okay, which are going to be formed, and they start occurring from the center, okay, towards the periphery. Sorry, this is the animal cell, but try to understand the concept here. The phragmoplasts are, they are nothing but the cell plates which are going to be forming the cell plates. It starts from the center of the cell and it moves towards the periphery. Okay, and these phragmoplasts, they are formed by three organelles. One is the Golgi complex, another is endoplasmic reticulum, and lastly the pectin containing vesicles. Okay, so this completes today's practical. Thank you.